thought your hand went up for a moment.
Oh, no.
Okay, so along that line, it gives me great pleasure to continue in Hammond's lab willingness to get out and do things first. Shelby has volunteered to give her presentation again this semester, even though this is still pretty early. So following in the footsteps of Kane, she'll be presenting her work today. Shelby joins us from the University of Arkansas at Fort Smith, correct? She has been a joy to work with so far and is really quite excited to talk about her simulation forest work and where we've been going with that. As it's always interesting to put a more human side to things. So as to humanize Shelby a little bit more than she already is, Shelby has a phobia of fireworks. And I think it is completely related that she really dislikes repetitive tapping. I think there's a connection between those two. So if you ever want to get under her skin, some fireworks and repetitive tapping is fine. So without any further words from me, Shelby.
Okay, everybody, thank you so much for joining me today. Like Dr. Kim said, I'm very excited and very zealous about this project and the goals that we're hoping to accomplish. So allow me to take you on a journey through the Garden of Eden. So the reason why we decided to focus on restoring the American chestnut is because back in 1904, chestnut blight was first recorded in New York. And it was a New York botanical garden zoologist that first noticed these canker sores on the American chestnuts. And by 1950, pretty much every American chestnut was infected with the fungus. And that means over a time of about 45 years, this blight fungus spread across the United States. So it was a very cosmopolitan parasitic fungus that decimated the species. And 3.5 billion trees were lost. And I just want to let you guys know that this is a very invasive fungus. It's not just something that's easily curable. It grows inside the vascular cambium of the tree, which if you take a tree and cut it in half and turn it over, you see the rings. On the inside, that's the vascular cambium that has the water in the xylem and the sugar in the phloem. And that sends that all throughout the tree so that the tree can stay nourished. And whenever the blight fungus is there, it disallows the tree from sending those water and nutrients up to the rest of the tree. And so in response to this, it's kind of like a human body. If we have a blood clot or an injury site, our prostaglandins and immunoglobulins go and they start to do scar tissue around that site. Well, that's basically the same thing that the vascular cambium does in response to the virus. It calcifies that layer into a canker sore. And because of that, they just pop up everywhere all over the tree like lesions, and they just take over and the tree dies. It like suffocates it and starves it to death. So the reason why the blight came in the first place to America is because we started importing Japanese chestnut trees, which are a cousin to the American chestnut tree. And the parasitica fungus was able to adapt accordingly to the American chestnut tree and just wiped all it. Even though there's still trees existing, it wiped out the survival of the species. So why reintroduce? So now we have back-crossed species of American chestnut trees with Chinese chestnut trees in order to create a viable offspring that can survive the blight infection. And we can see in this land trust right here, you see where there's no growth here and there's a lot of growth here? This is actually a land trust for American chestnut trees, showing people where the chestnuts were before they were decimated. So this is in Maryland, and all across this riparian zone, which is along this water line in this watershed, there used to be a ton of chestnut trees, and now there's none. And it just kind of gives you an indication that these trees were once really important and once really dominant. And so for us as humans to have invaded this land with a species that carried a virus over that would have normally not crossed the big lake without our intervention, we kind of need to take responsibility for that and at least try to get us back to what the land was before we influenced it so heavily. Also, American chestnut trees are thick, hardy hardwood tree. So they are able to sequester carbon better than a lot of other trees. So there's also like some effects with taking that carbon dioxide that we're accumulating so frequently in the air and sequestering inside these trees. And, you know, like I mentioned, these riparian zones, the American chestnut tree was found all across this water line. There's a reason for that. And that's because the American chestnut really likes those riparian zones. And because of climate change, because of our modifying ecosystems, these riparian zones are suffering. These are really delicate ecosystems that harbor really delicate animals like amphibians and certain bird species. And so anything that we can do to enhance those riparian zones is going to help the ecosystem and overall North American biodiversity. So I want to let you guys know what the 1516 American Chinese chestnut is, because the whole back crossing thing can be confusing. So I found a really good resource to explain it to you guys. What they did is they took a American chestnut, a 100% dying American chestnut, and they crossed it with a 100% healthy, virulent, blight susceptible Chinese chestnut. And that created a 50-50 first generation hybrid Chinese American. What they do then is take that baby, back cross it, which means breed it 3 times with a 100% American chestnut, giving that species the very close genetic genotype and phenotype of the American chestnut. And when they do this 3 times, they wind up getting a 93.75% American and 6.3% math Chinese chestnut, which means that the species is mostly American, which is really good. That's what we wanted to achieve in reassembling the species to be resistant against blight, was to resemble the American chestnut as much as possible. And so we have done this, and the American Chestnut Foundation is planting them in a lot of places. And this is the natural range of the American chestnut tree. A lot of replanting is going on throughout this whole area. But for our purposes, for our simulator, we had to choose a location to really zone in on and really focus on to see what the other species were, what was going on there, what the parameters were. There's a lot of consideration that goes into creating these models. And I'll get more into the models here in a second. I just really want you all to know the background. So what we did first is picked a site. And we looked at the site and thought, would this be a good place to reintroduce? And we found a ton of data at this place in Virginia called Beanfield Mountain. And the canal data set that we found mentions a ton of variables over time, such as growth in diameter and canopy, everything. So we were able to utilize that for our purposes in order to simulate our forest in VITA. And that's what VITA does, is we're able to take published data and assimilate it in a CSV and a YAML file that our simulator recognizes and is able to grow those species together in a world that we've also parameterized for. So we picked our Virginia spot mostly because we had good data for it. But I really want to emphasize that this is a great place, particularly for reintroduction, because in 2009, Dr. Fei, who is a very smart Chinese scientist, published a fantastic paper defining what the perfect parameters would be for the new American chestnut species. And she mentioned 3 major things. One was that the slope needed to be about 25 to 40 degrees. The second one is that the area could not be heavily influenced by agriculture, because that'd be stripping of the soil and then it wouldn't have access to the nutrients it needs or the water that would be found in the soil. And also, limestone cannot be found nearby the species either, because that messes with the soil pH, makes it more acidic. And so in our Virginia site, we have sandstone, we have a slope that changes from moderately steep to not steep, from 10 to 20 to 40, 50, 60. And then our land that we've picked is part of the Daniel Boone National Forest, so it's not been heavily influenced by agriculture. So altogether, Dr. Ham and I agreed that this Virginia site at Beanfield Mountain would be ideal for our purposes. Okay, so now we get into the good stuff. Here's VITA. So like I mentioned earlier, we are able to assimilate these species CSV and YAML files, which means that we parameterize our whole environment based on our species and our world. So we have to correctly parameterize both of these things in order to get satisfactory results. And in order to make sure we correctly parameterize, we hindcast and we try to replicate those forests that we have data for to make sure that our simulator produces the same results as a real life forest over time. And so a lot of our project consists of data gathering and comparative analysis, where we compare our simulated values to the published literature values and see how good they match up. So two things that we are thinking about doing are elevation and water access with geospatial modeling incorporated into our simulator. We haven't gone there yet, that's very technical, but that's something that I'm really looking forward to possibly incorporating. Okay, so what VITA does is it takes these files that we've been talking about, the CSV, YML files, and the world parameters, and it runs them all together. And then the output files are going to be a combination of that video that I just showed you on the previous slide, and then species-specific CSV files, and then the whole interactive species file over time. That shows you how each species at each year over time. And you get these in terms of pictures, CSV, video. There's a lot of outputs that you can choose from. And so VITA is one of those tools that you can use to represent data in a lot of different ways. And we have successfully used JMP as SAS product to review our variables in different ways and graph them. And it's been really successful so far. These are examples of our YAML files. YAML stands for yet another markup language. It is not code. What it is a recognizable typed parameter that the simulator recognizes. And they are able to, the simulator is able to take these as the foundation and then grow the species on top of those parameters. So some interesting things here for our world preferences that we use are the gravity's 9.81. If you've taken a basic physics class, you know why that exists. And then we also have allow random death right here. That means a seed has a 0.75% chance of dying randomly, which is absolutely around what we would see in nature. And I've looked that up in a lot of literature articles, and that is about the random rate of death. And then of course for our species files, there's a lot that we could do, but basically these YAML files lay the foundation for our simulator. And then our species files enhance that simulation. So as you can see here in the CSV file, we have these alpha and beta values. Those are species specific. And we are able to plug in those alpha and beta values here in order to reflect the dimensions that we would see for that species. So for woody diameter, canopy mass, to woody mass, for young and mature, all those things are important, and all those things need to be considered when parameterizing a tree species. This is what we shoot for. And although it seems like growth of anything should be constantly exponential, let's think about that. You grow really fast when you're a toddler, really fast. And then you get to like 30 and you're like, okay, I'm shrinking or I'm not growing at all. That's life. Same thing for a tree. Its woody mass, canopy mass, and a lot of other dimensions increase very exponentially at first, and then we see this tapering off effect. This point where we stop gaining exponential growth is the sign that your tree is sexually mature and able to reproduce. So that is the difference between juvenile growth and mature growth.
So these graphs in the CSV file allow us to graphically demonstrate if we would see this relationship based on the values that we edit here, our alpha and beta values. This, not so much what we want to see. But if we see here, that's what we want to see. And so you get instant gratification, like instant showing of whether or not you achieve what you want to achieve or not. Quickly, I want to go over our proof of principle with previous analyses using Vita. The first analysis done was by Dr. Hammond in 2009. This is published in the Journal of Botany. What he did is he parameterized for an Aves albus species in a heterogeneous angio and gymno environment. So that means he parameterized for a generalized conifer and a generalized angiosperm and grew it in a forest with Aves alba. And he used the Cannell data set published in 1982 that we talked about earlier. And these are all of their species-specific values. We're not going to get into that. And so what he did is, like I mentioned before, he compared what his results were to what the published values were, and he found that his alpha and beta values are statistically indistinguishable from the real-life values. So it was clear to him that this was a good product, that he should invest time in this. And then I was able to help him in the second analysis using Vita, where we looked at a homogeneous forest of Pinus ponderosa in Oregon, and we used some Ameriflex data. It was a compendium of a lot of different information. We were able to parameterize for the Pinus ponderosa, and we only looked at DBH of greater than 0.2 meters. And then our simulated values are in green, and those are averages over 4 trials with standard deviation. The literature values are in blue. And we found that our data was within a very acceptable percent of the published literature values. So we looked at this data, looked at the modeling, the number of trees, and it very much resembled that Oregon location. So again, we were solidified in our hopes of using Vita for analysis in the American chestnut tree. So just to recap the project, what it is, why we're doing it, you know about the chestnut tree, you know about Vita, here's how they go together. So there are tiny projects going on all over the United States of a seedling here, a seedling there, and just baby it and treat it preciously. Well, eventually, we're gonna have to do mass introduction of chestnut trees in order to resurrect the species. So we have picked that Virginia site as a possible location to reintroduce that 1516 Chinese American chestnut tree. And like we discussed earlier, that comes with correctly parameterizing that 1516 species, as well as correctly parameterizing forest and other environmental factors such as the Jansen-Connell index, which measures how far the seed is sent away from the parent as it's dispersed into the environment. If the seed is closer to the parent tree, it will be shaded, it will have greater competition for uptake of nutrients, and all kinds of other things that show ecological observations that are very important in these kind of analyses. So we are going to correctly parameterize those 3 things and also engage in several different reintroduction techniques in order to figure out which technique allows for the maximized success of our new species. So what we've done so far is figure out the top 10 most abundant species in our Virginia area. And using various resources, we were able to zone in on not only the top 10 abundant species, but over time, what have been the top 10 most abundant species. And let me assure you, those things do change. Species abundance does change over time. One species does not stay the most abundant forever. It's like everything else in life, it's constantly changing. And so we were able to pick these top 10 species, and the 3 most occurring were the white oak, northern red oak, and red maple. These are not in a particular order, but those are some of the three that we're going to be most centered around looking at. So, with our project so far, I've presented a brief discussion of the Pines Ponderosa analysis at the American Chestnut Foundation poster session in October. We got so much good feedback from people who don't know much about computers and technology. So it's a good sign that people who don't know what a YAML file is are still really interested in learning about this, because it does incorporate a lot of different dynamics of learning. And so what we're doing now is currently seeking for funding, and we're just kind of enhancing our simulator and making our way towards defining that best method of reproduction. I want to share with you guys my timeline just so you kind of know what I'm heading towards in the near future. So this month we have successfully done our first mission, which is gather land and species data for Virginia, and now we're parameterizing for all those 10 species. And then by May, I hope to start exploring and diagnosing those most effective reintroduction methods. By August, I hope to know that most important reintroduction method, as well as begin my process for publication and preparing for conferences. And then by November of 2019, a year from now, I want to prepare to defend my project, which just talking about it is really exciting. And of course, I'd like to acknowledge Dr. Hammond, Dr. Nicholas, in their developments of this simulator. It has been so much fun to work with, and it's taught me a lot about biology, ecology, computers, life. I could literally just like write a Bible on this thing. It's so crazy. And then also Ben Jarrett, who is allowing us access to the database, which like nobody has access to. That's like VIP only. And it's going to allow us to parameterize for our 1516 Chinese American Chinese chestnut. And here's my resources, and thank you so much.